Olfaction, in my opinion, and the mechanism at which signal is converted from an odorant to a chemical signal is probably one of the coolest signal transduction pathways that I can think of. And so what I want to do is I want to draw an olfactory neuron here. Right, and our olfactory neurons, right, stick into that olfactory epithelium with some epithelial cells. There's Bowman's glands, which produce the mucus, right? You produce a bunch of mucus inside of the olfactory epithelium, okay? And we're going to put a whole bunch of stuff on here, but the most important thing, right, is obviously there's going to be a receptor for a molecule that's going to predict or dictate the smell. So that molecule is known as an odorant. So this is going to be a receptor for a given odorant, okay? And so there are going to be a couple different molecules attached to our receptor. And so you may have guessed that this is a G protein linked receptor. And this G protein is the G protein of the olfactory system, which we aptly name Gulf. Okay? Then over here, we are going to put a molecule of adenylyl cyclase. And does everybody remember what adenylyl cyclase does? It converts ATP into molecules of cyclic AMP. All right, the other thing we're going to put on here is we are going to put another channel over here. Just going to draw it like this. And this is going to be a cation channel. And this cation channel is going to let in sodium. But it's also going to let in calcium, that divalent ion. And then way up here, let's do this in purple. We are going to put another channel. And this is going to be a chloride channel. Okay? And we are going to draw this chloride channel closed. Okay? So now let's take a look at what happens when an odorant comes in and it binds to this receptor. So we have the odorant, it's going to come in and it's going to bind to the receptor itself. What that's going to do is it's going to cause the G protein GOF to dissociate. It's going to come over here. It's going to interact with adenylyl cyclase, activate adenylyl cyclase, which will take ATP and convert it into molecules of cyclic AMP. Okay? Now, what cyclic AMP is going to do is it's going to come over here and it's going to bind to this cation channel. And when cyclic AMP binds to that cation channel, it's going to allow sodium and calcium into the cell. Now you're thinking to yourself, okay, that brought in some positively charged ions, which means that our membrane potential is going to go up. However, the amount of calcium and sodium that is let in is not enough to reach threshold and activate or generate an action potential in this cell. Okay? So what's going to happen is that calcium is going to act as a ligand and it's going to bind to this chloride channel. And when it does so, that chloride channel is going to open up. And chloride, a negatively charged ion, will then leave the cell. So we have a negatively charged ion, that chloride ion, leaving the cell. And so with that, this is going to cause the membrane potential to go up. And it's this step right here, that's the step that actually causes the generation of an action potential. Right? So it's not using voltage-gated channels, it's using ligand-gated channels, and the positively charged ions don't generate the action potential, they actually serve as ligands, and it's the negatively charged ion that generates the action potential. So this is a little bit different than what we see in traditional neurons, which is why I think one of the cooler signal transduction pathways.